from my side, welcome as well. As Leon said, today the focus is going to be on creating TVB digital twins, and actually the processing part of that allows us to get there to run our simulations. And the focus is going to be particularly on clinical MRI processing, so on a specific use case of ours where we have stroke data. So we're going to take a look at just generally how do we process MRI to make it ready for TVB. What is some of the challenge or some of the issues that we encounter when we talk about clinical MRI, in particular stroke, and then what we develop to deal with it, and what kind of research it enables us to do with TVB now that we have this, have dealt with the actual processing of clinical stroke MRI data. You already heard great talks about TVB, so I don't need to introduce that. Just as for the focus of today is basically on the first half of this, not so much on TVB itself, as that we see on the right. You're fairly familiar with what TVB does, and what we want to do with it is we want to have these mathematical models based on actual brain data, right? So we want to have these neural mass models for each of those regions. But to be able to do this, we need, first of all, to know what those regions are, where those regions are for our subjects, and in particular, how they are connected. So what we need to do for this is we need to have a parcellated brain, right? We need to know where the regions are. And then we need to have spectrography in most cases to get an idea of how well they are connected, these regions. So the combination of those two allows us to get our brain network model. So the question is, how do we get this from this, basically, right? We have a subject and a scanner, and how do we get to TVB? So this is what we're gonna try to answer today, in particular if that was happen to be a stroke patient, which is just a stock image. So I don't think that's the case, luckily. So the first chapter, like I said, is going to be just general MRI processing. I'm not going to go too deep into detail basically on anything, because I mean, MRI processing is a lifetime's work anyway. So feel free to, if you have any more questions about some details or anything, please feel free to ask them right away or at the end. Like I said, I'm open to both. But we're going to go somewhat of a high level description of some of those steps and how they actually influence processing. We will have, I hesitate calling this a hands-on session at the end, but I will go through some code that I also shared with you to maybe give a little bit more of a better understanding if some of you are totally new to actual MRI processing. So we will do this at the end as well, but first we want to go through some of these descriptions, which is going to be the main part of the lecture today. So as I mentioned, we have these different modalities, right, in MRI, and we need a combination of those to end up with our digital twin. The first thing that I said that we need is actually parcellation, right? So we need to get to a point where we have these regions separated that we know, okay, region 1, 2, 3, whatever they might be, and however granular they are, depending on the atlas that we're using here, right? We can have 40 brain regions, we can have 380 brain regions, which is what we have here. We can have 1,000 brain regions. This doesn't really matter right now, but we need to get to a point where we get these regions in the first place. And how we do this generally is we start with our structural imaging, our anatomical images of the brain. Right? On the left here, we see a T1 image, and here we see a T2, or in particular a FLAIR, a so-called FLAIR image, representing the same brain, but having a different focus of the actual properties that we capture, right? This is what differentiates these different MRI modalities. We're all always capturing the same brain, but we're capturing different aspects, different biophysical aspects, etc. And even within the anatomical scans, we have these different properties, with the most basic one generally being the T1 image, which gives us a good estimate of the actual structural properties. Generally, we then have a high resolution T2 image, but in our case, and most of the data that we see throughout this presentation will be just our stroke use case. We actually have layer images, which have the advantage that they are very susceptible to liquids in the brain, right? FLAIR stands for fluid attenuation inverse recovery rate. So it's generally used in the clinic if we talk about stroke patients to really get a good estimate and a good view of an actual lesion, right? This is what, if the patient comes into the clinic with stroke symptoms, we want to see where is the lesion, what type of lesion, etc. So this is where we have the focus here. And what we want to do is we want to start with this full MRI scan, right? Even containing the skull, containing all this in individual properties, and we want to end up with a fully processed image. I just highlighted this here as, I mean, the only thing that we really see here is that it is brain extracted. So we removed everything, skull, all organs around it, and just that we end up just with the brain. Of course, there's a lot of steps that actually go on, a lot of normalization, etc. But just to get from this raw image to our two processed T1 and T2 images, and we can then use this and the information that we can extract from there to create parcellations. Like I said, it's a lot more complex than this, but this is where we start. We start with these T1 images, T2 images, and we create parcellations. The same kind of approach is the case for diffusion imaging. We start with our raw DWI, diffusion weighted imaging. Again, same brain, but now it has the property of what is thought to be capturing the movement of water molecules in your brain, which again is interpreted as an approximation of how tracts are actually in our brain, right? Like if the chances of a molecule to go through your elements that we have, these volume elements, if the chances of it, the probability of it to go like from back and forth are very high, there's a high probability that maybe there's a bundle of tracks also going this direction as opposed to left to right, because we have these tracks going straight and not left to right. So there's a bit of the idea of how the probabilities are of tracks depending on the movement of the molecules that we track with diffusion imaging. And what this allows us to do then is to create these estimates of tracks that we have. And once we combine those with the already existing parcellation, we can then say, okay, for each of those regions, how connected is region 1 here with region 2, 3, 4? And we can create our structural connectome from this, right? So that we get a good estimate of the strength of the connection between these regions. And generally, we also get a good estimate of actually the distance between regions. So we get two structure connectomes that we want to use in TVB, one being the actual strength of the connection and the other one being the delay, basically. If we think about one brain region being active and the impact of that activity on another brain region will, of course, be delayed depending on how far away these regions are from each other. So we take these information from the diffusion imaging and integrate this into TVB. For functional MRI, we follow the same principle. We do a lot of pre-processing of MRI images, motion correction, distortion corrections, all kinds of corrections, basically trying to get to the actual signal underneath all that noise. And then once we do this, we can again use the parcellation, now shown here as these black ribbons, to see, okay, what is the actual activity in each of those regions? And once we have, for example, the average connectivity for each region, we can correlate them and then create a functional connectome, which basically looks the same. This is actually the same picture as I had before, the structure connectome, because our functional connectomes have a certain specific to it that I don't want to introduce yet, but we will see this later on. But it's basically the same idea, right? We get these same connectomes. They are, of course, highly correlated, but I don't want to go too much into this because that will open up the whole structure-functional relationship debate, which is in itself, of course, a whole field of research. And when we do this, we go to the next step. When we want to do this, there are already existing frameworks, right? It's not that in our project here, we invented processing of MRI. There are great processing pipelines out there, even multimodal ones, like, for example, the human connectome in a processing pipeline from Dasaya from 2013 creates high quality processing results and can be kind of considered the gold standard in automated processing of multimodal data. So if you have access to data from the Human Connectome project, that's basically the pipeline to go. If you want to process data yourself, they always publish a lot of pre-processed data already anyway. And then there are other pipelines, like, for example, fMRI prep, which also creates great processing results for fMRI data. Very specific to that modality, but creating very high quality outputs. But they all come with certain assumptions about the data. For example, Human Connectome project, I wish you would have data of that quality in the clinical context. But of course, this doesn't exist. So there are certain assumptions about the data to be able to use these pipelines. It's similar fMRI prep creates great fMRI data, but is somewhat susceptible to movement artifacts and the quality of the fMRI data itself. So there are always certain things that we have to keep in mind when we decide on using one of those processing pipelines. And what they all have in common, our pipeline as well, is that they kind of are a wrapper for a whole range of processing steps, right? So when we look at these pipelines, at the end we end up with our nice parcellation, but actually the steps that happen in between is an insane amount of steps, and a lot of them can be broken down to some of those very basic ones. The most basic probably be the actual registration of images. So if we want to align them, if we want to be two images to overlap, etc., we perform registration. We will go through all of them in a second, except for motion correction, because that is basically just registration applied to deal with a specific problem, such as motion of the subject in the scanner. Of course, if we think about resolutions of these scans of 1 millimeter cubes, most people that lay inside of an MRI will be not able to move less than 1 millimeter. So we have a movement around that. And we need to correct for this to make sure that each voxel actually stays at the same time over time to be able to compute the activity of that area. Others are surface reconstruction and segmentation, which we also have a slide or two to look into this real quick. And then artifact correction, which is kind of a broad term, which we will look at in more depth because of the lesion-specific artifacts that we deal with in our use case and in our pipeline. So there will be a little bit of more detail on that and on different things to that. So if we're talking about co-registration, like I mentioned, if we want to align images like we see here on the right, we have a T1 image of our subject, and we also have a parcellation, but we can already see that they do not align very well. Right, so if we want to make some inference about specific regions of the brain, we want to use an atlas, and we want it to be able to be used with that specific subject. Right, so what we need is this to actually align, like we see on the right here. We need those to overlap that we can say, oh, this region is actually representing the underlying brain, and not as we have it here. Of course, this frontal region is somewhere in subcortical areas and not actually at the front of the brain, and it's living in what is generally referred to as totally different spaces. The same goes if we have a template brain that we want to use. Like this could be a template atlas, for example, that exists in a template space, and we want to apply this to our data. We need them to be aligned, and we need to put them into the same space, and this is what co-registration does. And at its core, the idea is basically that we have an optimization problem. So we have two objects and we want to align them. So we define a cost function. This could be just the difference between the voxels that we see, right? We see a dark gray and we see a white voxel, and then there's a difference in the absolute value. Can be a little bit more complicated, of course. But we want to minimize this cost basically. So we want to make this we want to solve this optimization problem to have those aligned. We will get to this basic property several times throughout the talk today, I think. The other one is one of the basic ones is segmentation. So segmentation means if we look at our T1 image, and we now already talked about the gray matter and the white matter, and different where we track tracks and versus where we have our cortical regions in the gray matter, etc. So we want to actually be able to separate the different tissue types, for example, for downstream processing, for tractography. So we want to be able to get a volume or a mass that only contains our gray matter. Our gray matter ribbon right here. We want to have a mass that only contains the white matter. So we only have the tracks that actually connect the different regions. Or we want to have a mass for the cerebrospinal fluid. Also, again, for tractography later on, because we want to know where the brain actually ends. Right? If we think about following along a tract, we don't want to actually find a tract that goes deep into CSF, because we know there is actually no brain. We will not have actual axons, actual tracts there, because we're outside of the border of the cortex. So getting an accurate estimate of these different tissue types is also a crucial step in allowing these more complex downstream processing steps. And the last one, also, I would say one of the most complex one, and at least computationally expensive one, if we're actually running the processing, is the surface extraction. So if we look at our T1 image here, and if we zoom in, we already mentioned this, there's a 1 millimeter cubes, right? So we have a very pixelated view of the cortex. If we zoom out enough, it looks smooth. But if we actually zoom in and we want to have some idea about specific brain region here, specific cortical region, we get into the issue that, I mean, luckily our brain is not actually shaped like this, right? We don't have these cuts. So to get a better estimate of these regions, it would be nice to have a smooth representation of the actual surface of the brain, rather than having just these voxel cubes that tend to misrepresent distances, etc., in the actual surface. So the idea is to, instead of having these 3D cubes that we use to separate the brain, we map a triangular mesh onto the outside of the brain, as you can see here. So this triangular mesh is standard in computer vision, etc. So we see this, it's easy to map this and actually solve the optimization problem there to have a good representation of these triangular surfaces that allows for a very fine-grained alignment along the surface. So we kind of get away from this 3D clunky representation of the voxels into a smooth overlay of the actual surface, which kind of helps us solving some of these limitations of the spatial approach, this volume approach. For example, if we just think about, we want to know how far specific regions are away from each other, we can think of this as computing the distance between two voxels. If we now are still in voxel space, we have this voxel representing the gyres over here and over here. And then if we compute the distance, it's just going to be very inaccurate estimate, because we're voxel-wise, we are very close to each other. But if we look at the truth, of course, those are actually far away, because there are no connections in here. So the connection going from this region to the other region is actually fairly long. And what the surface representation allows us is, instead of going through exactly one voxel to get to the voxel of our target region, we're actually walking through a whole range of triangles that we have to pass into this mesh, in this grid of the surface, to actually get to our target. So we get a better estimate of the actual distances. And I mean, a lot of other aspects, but this is trying to get to these advances of surface representations, as opposed to just these 3D representations of the volume themselves. And if we take all of these steps and a lot more together, we end up with these processing pipelines. So example here for the human connectome processing pipeline, we see that those are organized in different modules. In this case, it's pre-free surfer, free surfer, and post-free surfer, and also the fMRI volume processing pipeline. And we can see that those are, again, subdivided in specific steps, right? We start here with our original T1 and T2 images and our field maps, and then we perform all of these different steps. And then we see, for example, alignment and averaging, right? So this box in itself is already multiple steps of registration between the T1 and the T2 image to get them into the same space. And then if we have, for example, multiple T1 images, which can be the case, we want to make sure that even the T1 images are in the same space, right? Because even between those data acquisition protocols, we might have movements, right? So we might have these slight differences. And if we're talking about these very high resolution images, that might make a huge difference. So we want to align them, multiple registration steps, and then we have an ACPC alignment. So we have that long anatomical landmarks in the brain that we want to align to. So there's a lot of steps that actually make up a single step of such a volume. Those 4 are just selected because those are the ones that we're going to keep for our actual stroke pipeline, but we don't need to go into too many different details here. So in the end, we have these pipelines to process diffusion MRI, anatomical MRI, and also BOLT, so fMRI signals, to create TVB-rated data, right? Misha Schöner, for example, from our lab, published a pipeline here in 2015 to create this data. There's the human connectome paper pipeline. There's a range of pipelines that can either do all of this together or just specific modalities to create our structure connectivity. And I would say in most applications of TVB, ideally either functional connectivity or some measure of the functional activity in such a way that we can fit our models and we can compare our models to some empirical ground truth. This graphic is actually adjusted because we also have EEG data here in the original publication, but we're not focusing on EEG today, just on the MRI, because EEG will be a whole other level of complexity and talking about artifacts. So now that we know how to just generally process MRI data, we have the pleasure of looking at the challenges of the ischemic stroke, which is the basis for our use case. We have some data from our collaborators at the Universitätsklinik Hamburg-Eppendorf in Hamburg that collected this data over several years, nice longitudinal data. And the goal here was to use TVB to solve some of these issues because, well, stroke is one of the major causes of disability and also death worldwide, and particularly ischemic stroke. It happens rarely often. We still don't really have a good biomarker for recovery prediction or for treatment options to have a clear biomarker for deciding on treatment option. There's a lot of ambiguity in terms of biomarkers for this in the stroke community. So the goal was to kind of establish this and kind of expand this using brain network models. And if we want to kind of categorize the problems that stroke causes for the MRI or for the field in general, we can kind of categorize those into those 3. The first one being data. The challenge comes with the data, already with the data itself. And in particular, we know that stroke, and we see this in a second, has a huge dynamic over time. So the typical stroke patient, I mean, first of all, this doesn't exist because of the high variance within the population. But if we take a look at the stroke in a patient while it's happening 3 days after, a week, a month, a year later, it will be a different stroke manifestation each time. There's a lot of dynamics. So what we want to have, especially to enable recovery prediction or the treatment options, we want to have the acute phase. Like we want to know as soon as possible the manifestation of the actual stroke. But as we're talking about patient data here, we will only find this data in the clinical context, right? If you have a stroke, you represent yourself to the emergency room or some other place, either way in the hospital. The actual data acquisition generally takes place in the hospital as well, and there are specific limitations. For example, if we think about the actual timing for data acquisition, right? I mean, in stroke, there's a saying, time equals brain. So we do not necessarily have the time to run these extensive data acquisition protocols as we do in research, where we just scan healthy students that have a lot of time to actually spend in the scanner, as opposed to the basically terrified stroke patient. So this already limits us in terms of times, but also in terms of availability. If we're talking about the HCP, where we have 7 Tesla imaging, as opposed to clinics where we maybe these days have 3 Tesla if we're lucky. So just the type of data that can actually be acquired already differs significantly to what is used in, for example, creating HCP or fMRI prep pipelines. The other thing I mentioned already is the actual variance in the disease. So we, even in our use case, all of our patients represented with hand motor deficits. But this does not mean that they all have a similar lesion, right? So even the actual pathology that we see for a disease has a huge variance within ischemic stroke patients with the exact same symptoms. So this is already a problem that we might find a solution for one, but not for the others, right? Disease represents we have this challenge in actually trying to find mitigation measures to combat this. And the most, I'd say the most impactful one is that the data itself, the acquired data, the lesion information in that data presents a challenge. So we can think of these high quality, high resolution, nice T1 images used in the Human Connectome project, and then can think of a lower resolution image from a clinical context where half the hemisphere is covered by a lesion. So of course, this will present a challenge to existing frameworks in trying to get information about the brain if now it's not actually looking at the full brain, but there's actually a lot of damage, a lot of abnormality in the data. So these methods might not be sufficient anymore. And if we just to look at some examples for this, we talked about the temporal dynamic. We see this here with a large right hemisphere lesion, subcortical cortical lesion for this patient. If we then look at the exact same patient 3 months later, the lesion represents very differently, right? So not only do we have these variants across patients, even though they have the same symptoms and how the lesion manifests, even the lesion itself within a patient, this temporal dynamic changes dramatically. So finding a solution for a chronic lesion, as we have here for the small subcortical area, might be easier than finding a solution for this large lesion that we see here that represents strong abnormal intensity values compared to the rest of the brain, right? So we need to be able to find a solution that kind of works with even this very variant data that we have within patients, across patient populations. This is some of these challenges that stroke themselves represent. And to look at an example of just what I mentioned about the quality and the resolution of the data, at the top we see a nice high resolution T2 image from the Human Connectome project. It's beautiful. The resolution is very nice. The segmentations that can be done on this is very nice, high contrast, high resolution. And then we compare this to the equivalent in our data set that we have, which is not a T2 weighted, but an actual flare image. We can see that there's not only the issue of the actual lesion that we can see here, but just the resolution itself, right? We see a significant decrease in the actual resolution of the data that we have available. And the same is true for fMRI data. At the top here, we see the nice fMRI task and resting state data from the Human Connectome project. And at the bottom, we see the data from our cohort, where not only do we see a reduced resolution, but also we see this issue at the bottom here. This is not that I cropped the image, but this is the actual image where we have large parts of the brain actually not acquired, right? As I mentioned, the selection criteria for our data set was a motor hand deficit. So the focus during this clinical acquisition of functional MRI data, which is also very rare to begin with, to actually do fMRI in the clinics, the focus was on just capturing the motor cortexes and not so much for capturing the whole brain. So what this resulted in is that basically, unfortunately, for all of our patients, we have a lot of the brain actually being cut off. So we have a lot of temporal areas, a lot of the occipital areas that are actually missing from our brain. We do not have data for this. So if we now think about established processing pipelines that tend to take a full brain and try to process this, we can see that maybe we can have some challenges in trying to actually process the data that we have from this clinical stroke patients. And here just a little bit to kind of bring up the point of the variance between the subjects. So these 4 plots are 4 different subjects. And on the x-axis, we see the 383 regions that we have in our postulation. So those are all after we process them. And we see all of them represented with the same hand motor deficit. So it's similar severity and similar deficits regarding hand motor coordination. And we see in black at time point 1 the actual impact on a specific region. Right? So if the black bar here is very high, it's 2-1, which means that this region is entirely encapsulated by the lesion itself. Right? So it's fully engulfed in the lesion. And we see already just the sheer number of regions that are affected differs significantly. We have on the left here, left hemisphere, right hemisphere, and then subcortical areas. That varies significantly. Here we have some left area, here we have some right hemisphere, we have subcortical areas. And then from black to blue to green to yellow represents the 4 time points, so that we see over time a lot of the black, a lot of these areas actually disappear. And we have different areas coming up because we have, as we've seen on the slide before, this dynamic over time, the lesions. So we have now specific regions that are not actually affected anymore after some recovery of the lesion manifestation. And the idea here is basically introduced by Burris in 2015 and also by Fox in 2018, is that we now don't look at the actual location of the lesion anymore, but we look at the affected networks. So those is just an example of 3 different patients that have the lesion at a different location, but represent with the same symptoms. And then if we take a look below, and if we take a look, for example, at the actual connectivity, we see that these regions are actually highly connected. So the damage that this lesion causes is not necessarily just the local damage, but it's actually to the network. And the same we can do for functional connectivity, where you can say, okay, those networks are actually highly correlated. So there's ongoing activity and interaction between these regions. And if one of them is actually damaged, this might disrupt these functional networks. So really the idea is to get to accurate representation of these underlying structure, like the functional connectivity and the anatomical connectivity, to get a better idea of the actual lesion impact on the subject, as opposed to just looking at a specific region that is affected or a specific location of the lesion itself. And I think this is basically just another representation of the lesion that we have. So if we think about this, this is from a second data set that we created, which I will go to later. But if we think about the actual abnormality and how these lesions manifest in the brain, this now is the actual healthy brain that is visible. And if we look at this area right here, we have a nice contrast of the cortex, of the subcortical, the tracts here, the white matter. And if we now look at the same brain but with a lesion, we can already see a lot of distortions. Right? So just as a representation of how these lesions look. Now, one of the main issues basically that we end up with is that now we have like a simplified representation basically. is we have dark gray areas where there should be just light gray, right? So we have basically some intensity values that we usually found in the cortex and the gray matter area that are now represented in white matter. So if we think about all of these volumes that we have as just 3D matrices or 4D matrices over time, and if we think about processing algorithms working on these matrices, we're now talking about just changes in the value at specific positions where there's usually a high intensity value, because we're talking about white matter. All of a sudden we have dark gray areas that are represented at the spot where there should be white matter, right? So if we're thinking about, for example, looking for specific landmarks in the brain, those might be distorted because we don't find a specific landmark there anymore because it's now shifted or distorted in terms of the intensity value. And if we actually look at the distributions of the intensity values, we could see this really clearly. So what we did here was we extracted, for all of our subjects, we extracted all the lesions, and then we plotted the distribution of the intensity values that we find there. And then we did the same thing, not at the lesion location, but at the exact same location in the contra-lesional hemisphere. So we swapped the dimensionality of the brain, and we took the exact same location in the other hemisphere and then looked at the actual distribution of the intensity values. And what we see here is that for the healthy signal, we have a clean unimodal distribution here of our intensity values. And as opposed to the lesion signal where we have a way flatter, way higher skewness of our intensity values. So what this means is that while in a lot of these regions we have a fairly clear distinction between intensity values, so we have a high contrast, right? We have these high intensity values, a high distribution of these high intensity values in our subcortical areas. Those are significantly flattened in the lesion area, which means that we have a lot more variance in intensity values in our lesion as opposed to healthy signal. So if we're thinking about actually looking at the gradients between gray matter and white matter to find, for example, the surface and find these different tissue types, we now end up with a problem because we don't have these clear cuts between gray and white anymore, because everything is kind of smooshed out in the lesion signal. So to kind of deal with this and try to overcome it, we developed the lesion area automated processing pipeline called LEAP, where we try to address all of these, or most of these challenges that lesion data represents. So the idea is, as I mentioned, we have this clinical stroke MRI data, and the idea was to enable the processing of these data to be directly ingested into the virtual brain, so that we can use brain network modeling to, for example, find biomarkers for recovery prediction. And the approach here was to create data that is robust, or to create a data processing pipeline that is robust, validated, and automated to really enable reproducibility, to make sure that what is computing at the end is actually representative of what we know and what we want the data to produce, and that it is highly automated so that it can easily applied to other data sets, for other researchers to use to kind of solve this reproducibility issue that we have, in particular in stroke data processing, because everybody just uses whatever pipeline, and either they perform corrections or they do not actually perform corrections, because a lot of times, especially if there's a smaller lesions, processing pipelines will still work. They will just give us distorted results. And there is a little bit of a lack of communication about what is actually, if we perform corrections, what are those and how are they especially implemented. And then it's, of course, always a breeding ground for non-reproducible data. So to kind of solve this, we try to get to an automated, reproducible, and validated pipeline that can be used to run TVB, but also just to process this data in the first place, right? Not everybody wants to do brain network modeling, but still there's a lot of MRI-based stroke research that is not adequately dealing with these lesions during the processing themselves. So we're gonna take a little bit look at the methods that we try to implement to enable this, basically in the categories of processing and reproducibility. First, the processing itself, we're going to look at the cost function masking as a way of trying to deal with the lesion impact, virtual brain transplants to enable a lot of downstream processing that is otherwise maybe not possible because of these lesion artifacts that we see, and pathological tissue tractography to kind of get a good estimate of how the lesion actually impacts our tractography when we want to create these structural connectors, right? Because we want to make sure that the integration of lesion into to this lesion-aware processing is accurate and gives us a good estimate of the underlying ground truth. And on the other side, reproducibility, as I mentioned before, because we want to make sure that the data processing is reproducible and usable by other researchers and other data sets. And here, the main aspects are containerization. So we already see the logo here, Docker. We used to create a Docker container for this, and also standardization, particular bit standards that we used. But we're going to go through all of those 5 measures next. But again, not in too much detail, but just to give some idea of what is actually behind these approaches. So cost function masking has been around for some time. I mean, the paper introduced in 2001. It's ancient in terms of MRI processing. And the idea basically is, we talked about this optimization problem before to align images, right? And if we think about, for example, we have this lesion patient right here, and we want to register this image to a template space, right? MNI space, because we have an atlas that we want to use, whatever. And if we then just use normal registration, that optimization problem treats the whole brain as its input and the whole template as its reference. So it's trying to map these two. And what we can see is here, if we don't mask it, it's trying to actually fill in the space that is not present in the template because that is based on healthy subjects. It's trying to warp what it can find of the brain and to reduce this difference, right? Because we said it's an optimization problem, so it's kind of warping all of those voxels inside, giving us at the end a registration to a template space. But of course, it's not accurate anymore, because then if we want to infer something about the lesion, this lesion is not really representative of the underlying true lesion that this subject presented with, because we can see that the sheer topology of the lesion changes significantly. So the idea is to do the optimization problem, to find a solution to it, but to only do it on the healthy tissue that we find. So that's the idea where the masking comes in, cost function masking. So what we give as an input to the actual optimization problem is then only all the voxels that are not covered by a mask over this lesion. So we say, solve this optimization problem based on everything that is healthy, and then afterwards apply the solution to this optimization, to this reduced optimization problem, to all the voxels, so that we can at the end find a registration in space, in template space, and still have a very good representation of the true topology of the lesion. And if you compare these two images, they're fairly similar, even after registration. That's the idea of, because we've seen this before, we have a whole range of registration steps all across the processing pipelines. And even if they're not as extreme as this, we have these slight differences that we might encounter. Those just grow and grow each time this happens. So we really carry on slight differences and slight errors across our processing pipelines if we do not pay attention to these small details, for example, with cost function masking. The other one is a bit more complex. It's based on the idea of enantiomorphic normalization. So basically, you take this assumption a bit to the extreme of the symmetry of the brain, and you say, well, I have this damaged hemisphere, but I know how the other side looks, how the other hemisphere looks. Sorry. So I can just use that information to guide to an estimate of the underlying ground truth that we can find here. So the idea is that we remove the lesion and we replace it with what we find in the other hemisphere, so that we can have this estimate, this approximation of the actual ground truth, to then be able to do downstream processing. For example, free surfer, if we want to extract the surface, we've talked about this before, right? We're trying to map this triangular mesh onto the outside of the brain, and then that is always just defined by the intensity values that we find, right? Outside of the brain is basically black, CSF. So if you're trying to map this in, we still find very dark spaces here that could be misclassified as CSF, as outside of the brain. So it will treat these subcortical areas as surface, even though they're only because they're very dark, even though they're subcortical areas, which is of course not the surface of the brain. So with this approach, we can kind of get an approximate of the actual underlying truth that we cannot get to because of the lesion. And the way that we do this is, because we use this idea of symmetry, is we basically take our input image and then we mirror it. And we can already see here the tilted lines in the middle, which means everybody lays inside of the subject, inside of the scanner, a little bit different. Our brains are, of course, not perfectly aligned in the middle. But if we want to use this idea of symmetry, we need it to be perfectly aligned in the midline to be able to actually swap the sides. So the first step is we mirror the actual image, and then we register this image to the mirror image. So basically what we're doing is we're kind of solving for how do we get from this left tilted image to the right tilted image, and what is the warp that we need to apply here. And then we cut this warp in half and say only now give us half of the transformation. So going from left tilted to right tilted, and half of this is basically perfectly midline aligned. And then we see this midline aligned image. And then we can use the idea of actually just swapping alongside this axis and get the same area. So we then use this lesion mask, we smooth it a little bit to allow for a little bit of the surrounding voxels to be included. And then we use the smooth lesion mask 1 to remove the actual lesion from the input image, as you see here. and two, to get the healthy signal from the mirrored image, right? So basically from this image that is now midline aligned, we apply the same lesion mask, and instead of taking out the lesion, we take out the healthy signal and then combine those two together, again, perform this half transformation into the left tilted axis, and then we get the approximation of the underlying ground truth of exactly the same space from this image and this image. So we have a close approximation of how this brain would look without the actual stroke, so that we can process it. And the other mitigation measure and processing that we did is because, as I said, we want to make sure that the tracks that we get, these connectons that we get, actually represent lesion information as well. So what we do here is we generally perform what is called anatomically constrained tractography. What I mentioned before, for example, if we have this tissue segmentation, we have cerebrospinal fluid, we know that we don't have these tracks inside of cerebrospinal fluid. So the algorithm is only keeping tracks that are randomly created based on these probability distributions that we've seen before. As I mentioned, the diffusion-weighted imaging, but we have this probability for each of those voxels. If we come from behind and we go through, or we come from the left, basically in 3D space, what is the probability? And it just creates millions of tracks randomly and then selects those that fulfill this criteria of not going through CSF, etc. And we can here also introduce the lesion as pathological tissue and say, well, actually, this tissue is damaged. So the tracks that go through here, also discard them, because we basically treat this to a degree as CSF, where we say, oh, this is actually damaged tissue. We do not follow along these tracks because we believe that those are damaged. So to get our connectome to represent these damaged tracks as well, we introduce this in this anatomic constraint photography to get a better estimate of the impact of the lesion on the actual tractogram, and therefore then on the structural connectome. And for the reproducibility part, as I mentioned, the Docker created a Docker container, but the basic idea of Docker containers that will contained in the name itself is that it is a container, a self-reliant container where everything that you require to perform within this application is contained in this Docker container. So that if you want to be able to run it, all you need to do is to be able to run any container, as you've seen here, and then all the software that is actually required to do whatever is happening inside the application is already contained in this. There's no additional installation. There is no dealing with dependencies between packages, conda environments, whatsoever. Everything is installed already inside of this container. You can see this a little bit as a virtual machine. There's a lot of differences, but just as an idea, it's like a self-contained. It has an operating system, and has all the software installed that you need, and then you just call it via the Docker container. What is really nice with this is that this approach also makes it platform independent. So whatever your infrastructure is, whether that's your local laptop or that's a supercomputer, doesn't matter. There will be one operating system or the other on that software, on that infrastructure, and then all you need is to have a containerization software, for example, Docker or also Singularity. That's the only requirement that you have, and then once you have this, you can run every application that uses Docker containers on that actual environment, independent of the actual infrastructure or hardware. Generally, those are very Docker is very nice in terms of using hardware efficiently, or also Singularity. So that is the idea, to kind of make it reproducible and to be able to easily share it. For example, we also have this TVB run container. So if you have Docker, all you need to do, if you want to have TVB and you don't want to deal with the different installation problems, etc., and you want to be able to use the exact same running environment as your coworker, you just pull the container, saying Docker pull, and it will automatically download an image of this container into your runtime, to your Docker container software, and you will be able to run it. We will do this later on in the example as well. And the other aspects that we mentioned for standardization, for reproducibility, is standardization. And what we did here is the idea that if the data follows a certain standard, we will be able to make this pipeline usable for any other data set that follows the exact same standard. And the main standard here is basically the de facto standard is the BIT standard, the brain imaging data structure standard, which comes from new imaging data, particularly MRI data, but it's community driven. So it now has been extended to a whole range of modalities, to include behavioral data, to include MET, EEG data, a lot of basically almost everything that we encounter in this type of research fields is now being covered by BITS. So there are these extension proposals. So the ideas for our LEAP pipeline is that the data that we put in, that we see here, the input data is following BIT standards, and also the output data that we create is following BIT standards. And what this means is that, for example, how the data points in the subject are named follow the specific pattern. So that it always starts with a sub, dash, and then the actual ID of the subject. And then within we have sessions. Those can be different trials that you run. In our case, those are the longitudinal sessions. Session 1 be acute phase, 3 to 5 post-stroke onset, 1, 2, 3, 4 is 1 year later. So we have within the subject, we have all the different sessions. And within each session, we have the differentiation between the anatomical scans. In our case, this is the T1 image and the FLAIR, the fusion scans in a specific format with adjacent the BVAL, the VVAC files, the functional data where our fMRI, our bold data is, and the corresponding event files, and field maps. And in the same way, our output data is following the so-called BITS computational modeling extension proposal, which has been developed in our lab by Patrik and Michael Schirner as well, which kind of tries to follow this BITS standardization approach for data that can directly be ingested into brain network modeling, in particular the TBB. So what we have here is we have the folder for the time series data, where we have the processed average time series for all our regions, which we want to use to fit our simulations against. We have the network folder that contains the structural length and weight matrix, and also our functional connector. We have a coordinates folder that contains the 3D coordinates of the centers for each of our regions. And in our case, we also added a T1 image, a T1 folder with the final T1 image, and also the individual parcellation that we created, and also a lesion folder, which contains information about the lesion load for each specific region of interest. So we computed, I think I mentioned this before, we computed these lesion load metrics that for each region of interest gives us a value between 0 and 100%, basically saying how many voxels of that regions are actually part of the lesion of that region, and actually part of that lesion itself, to give us some information, some influence about the lesion that we then want to use to model in TVB, for example. So what we ended up with is this multi-model automated framework, which is standardized as a BITS application. So each BITS data set can be directly run with this processing pipeline. It is compatible to clinical data. We've seen this just by looking at the resolution and looking at its reduced field of use, etc., for fMRI data. And then we incorporated all these different mitigation measures to actually deal with the lesions themselves. Just as a quick overview for the 3 main modules, the structural processing, we first run the step of cost mask creation. So you see all of those striped ones are new implementation, and the blank ones are adjusted existing pipeline scripts. So here we create these cost masks, because as we mentioned, there are a bunch of registration steps happening, and for each of those, we want to make sure that we introduce cost function masking, right? We want to reduce the lesion impact on these processing steps, as we discussed before. So we first need to actually create all these lesion masks for the different modalities. We need one initial input lesion mask that's usually has been done already in the clinic, where people draw the lesion based on the T1 image or the FLAIR or a combination of any of these. And then we create those for the different spaces that we register to in the different resolutions. Then we perform the anatomic brain correction, so the mature brain transplant, to get to this estimate of the ground truth. Then we can use an extended and adjusted version of the HPC processing pipeline, because as you said, the goal was to use the gold standard and make it so that it can process stroke data to not lose any of the quality of the actual processing. We want to go with the gold standard here. And then at the end, we have, again, individual parcellation mapping, that we get these nice overlay, these nice parcellations for each individual subject that we can then use down the line for the next processing steps. For example, for the diffusion processing here, for example, we had to implement a new version for the full pipeline, because the data quality did not even provide the required data to use the processing that has been used by the HCP. It's not just that the resolution wasn't there, it's just that the actual extent of the data was not sufficient. So generally, what you now do in high quality diffusion data acquisition is you acquire the diffusion data when scanning in one direction, and then you introduce some distortions because of the way the magnetic field moves through the brain. And then you acquire the same data, but now you inverse the phase encoding direction. So now you have the exact same distortion in the opposing direction, and then you can use the idea of these diverging directions, reverse directions, actually canceling each other out, so that at the end you get a nice clean approximation of the actual data. We don't even have this type of data, so we weren't able to use this. So we used our own implementation based on the Matrix 3 processing pipelines that exist, and adjusted these for stroke again, just running a lot of pre-processing steps such as distortion correction, bias correction, motion correction, etc. Running intensity normalization across the population to make these intensity values comparable across subjects, so that a light spot here means the same across all the subject, and we don't actually have these impact of individual variability. Again, we do the segmentation to enable us to perform anatomically constrained spectrography. We're averaging the response function, again, to allow group-based comparison. And then we run this anatomically constrained spectrography and sift to get from these tracks that we've seen with the combination of the region of interest that we have to create our connectome. And in this similar fashion, we start with our functional MRI processing. When we start with the raw data, we directly use the adjusted HCP functional volume processing pipeline, but then need to perform our own steps with the rest, because we cannot use the rest of the HCP for this. So we have new implementations again to register this data into T1 space to allow us to again use the parcellation to then compute for each of those regions the average time series, and then parcellate those, correlate those to get the actual correlation between us and get these functional connectivity matrices. And this is now an actual functional connectivity matrix. As I mentioned before in the beginning, I showed the image of the structural, because here we see these white columns and rows, which is not that something was not correctly computed, but it's rather those are artifacts from what you've seen before, where we have these areas of the brain that are actually not covered. So we actually don't have the time series data of these regions. So the correlation, of course, is zero, because there's no time series. So we end up with these large parts of the functional connectivity matrix that is actually empty, because we do not have these regions, which again proposes a little bit of an issue if we then perform, if we want to do some inference and analysis regarding stroke biomarkers on this data. We already started with the problem of the raw data not providing everything, and then it just keeps being carried through all the way to the downstream analysis and processing steps. And then to show a little bit how we approach the containerization, a lot of documentation with corresponding GitHubs. But basically what this allows us then to do is that if you want to use the software, you can either clone the GitHub repository and then go inside the repository. And all you need to do is just say Docker build from this Docker file. The Docker file is basically a cooking recipe to build the container yourself. Or you can directly pull the container, as you see with the TVB example, the Docker pull, and then the corresponding address where you find it. And then in the same way, it's very straightforward to actually run the container. If you just say Docker run leave processing, will just run the processing, but there's no input data given, so it doesn't know what to do. It will just give you the help message. And then to actually run some processing, the example down here gives us the three main variables that we need to provide. So if we want to run the processing itself, we first need to mount a volume, so the study folder that we have, where our data is stored according to the bit standard, and then we mount it inside of the container and just call it data. You can call it whatever we want, but then we would have to define it later on. This is just default values. And then we have the variable, which is probably the most important one, which is called steps. And here we just have a space-separated string that tells the container what processing steps you want to run. In this case, what we want to do is, we've seen this before, this module pre-free surfer. We want to run this module of the structural processing pipeline, and we want to include all of the lesion-specific mitigation measures, right? We have the ability to not include those if we, for example, process healthy control data. We don't want to perform virtual brain transplant because there is no lesion to transplant. So we have the ability to define this here. And then we provide the ID. As you've seen before, bits tells us that all of those will be, all of the names will start SUB-, and then this ID will be added. So subject P001 will be processed here. And then we give the name of the actual container. Here we see the overview of the actual granular steps that we can perform. The most easiest one is if we say steps and we say full, it will run the whole pipeline. But we can break this down, as you see, for example, here to pre-free surfer, post-free surfer, just creating the parcellation maps, just creating the tissue segmentation during the diffusion processing, etc. So in total, I think those were, if I remember correctly, I created like 27 modules that can be called like this directly, or just all together, either all of them or within the modality. So if you want to perform all the structure, we just say structure and all. And the container will automatically run this because it knows where all the data has to be, because it's following bit standard. So what we end up with is we have our data that we processed, we get a nice parcellation, we get the integration of lesion information that we see here, and still we are able to process our data. We get our true structure connector, as I mentioned, the weights, the strength of the actual connection, and the distance that the information between these regions has to travel, the functional connector, and some information about the lesion. As you see here, the left hemisphere A1 region is impacted by the lesion by 4.11%. So this is some information that we can introduce to TVB when we're actually doing the modeling, which we will see later as well. So now that we created the pipeline, we also want to make sure that it's actually doing what we are hoping it's doing. So we try to come up with a way to validate the actual processing. And the idea here was kind of the established way also in, for example, lesion segmentation, automated lesion segmentations, and done a lot of lately in papers with trying to use deep learning to automatically find such a lesion in the brain, for example. The idea there is that if you need a ground truth to compare it to, so you take a healthy brain and then you embed an artificial lesion. Generally, those have been just like black blobs or spheres that have been introduced. But if you look at them as a human being, you instantly identify the lesion because it is artificial. And the idea we had here is that we don't want to be able to just deal with lesions that we know are not representative of the actual patients that we see. Because if we are instantly able to differentiate them, there's some reasons for that, right? Because they are very pixely, they are very or too round for an actual lesion. So this our results may then be able to process these simplified versions of lesions, but not the actual ones. So the idea here was we take at the top row, we take a healthy control that we have age-matched in our data set. And then at the bottom, we take an actual patient. You see here, there's a patient with a stroke lesion right here. And then we take this lesion and we put it inside of our patient, inside of our healthy control. So what we end up with is this artificial lesion brain. And we see here this lesion embedded in our healthy control, and it is visually, in simplified version that we have now, it's not directly identifiable as being artificial. It is very close to actual representation of these lesions, while still being able to provide us with ground truth, because we have the actual ground truth of the healthy control. So we created a lot of these data sets by combining our healthy control with 15 randomly selected patients from our data set to embed those into the healthy control. And then we performed a lot of visual quality control to make sure that at the end, the data sets that we select are not directly identifiable as artificial. So we have a fairly complex algorithm of actually embedding this data, but if we're thinking about, for example, a very small in volume-wise brain, and then the lesion is taken from a patient with a very large skull and a very large brain, the simple size differences can end up with lesions maybe going all the way into ventricles or into the skull, etc., which of course is not realistic because we know we don't have a lesion in the actual skull. So we removed all of this data where this random combination of healthy control on the patient ended up with an artificial lesion that was directly identifiable as artificial, and we ended up with a total of 82 artificial lesion breaks that we can then use to perform validation of our pipeline. So we ended up with these two data sets, the ground truth, the actual healthy controls that we have that are healthy, and these artificial lesion patients, the 82 where we embedded the lesion inside of the healthy control. And then we processed all of those, the ground truth we processed, that we then call leap controls, because that's the ground truth that we want to compare against. And then the artificial lesion patients, we processed one with our LEAP pipeline, including the lesion mitigation measures, and then without the lesion mitigation measures, which basically means we don't use the adjusted forms of these HCP scripts anymore. We use the basic HCP structural minimal processing pipeline that already existed before, that is not lesion aware, quote unquote. And then we have the parcellation for each of those three processing results, and we started comparing those. So we computed agreement metrics between the parcellation, so for the full brain, and also for each region of interest individually. And then we did the same thing, comparing the ground truth with the results from the HCP pipeline that we get. Then using this parcellations, we also ran tractography again, and we computed the structural connectomes, computed graph and network metrics on these structural connectomes to then be able to compare them and say, okay, how similar are the graph metrics from the processing results from LEAP with the ground truth, and how similar are the processing results and the graph metrics of those from the HCP and the ground truth. And then at the end, we could use these agreement metrics to run some statistics and see, do we actually get a difference in reconstructing this ground truth using the LEAP versus the HCP? Are they similar? Are they significant, etc.? So what we end up with is we have our artificial lesion patient right here. It's actually a healthy control where we embedded this artificial lesion. And then we have in green our ground truth parcellation mask here now just binarized. So these are all the regions, all the cortical and subcortical regions, the mask for those, and the ground truth that we know to be accurate. And then we get the same mask based on processing with the HCP and based on processing with LEAP. And we can directly see in the HCP, where we do not have these lesion mitigation measures, we exactly get into this issue that I mentioned before, where now these subcortical lesions actually treated as cortical regions. Because of these abnormality and intensity values, it's misclassifying voxels here as being part of those cortical foldings that we see in the actual ground truth here, but that is distorted here in the artificial lesion brain. And then we see, after we did the lesion correction, we see the extracted cortical ribbon here based on LEAP, and we see this is significantly, just visually now, comparing those to the ground truth, we see that this reconstructing ground truth seems to be working fairly well for LEAP, but it's not working very well for the HCP, because of course we have this distortion of the lesion itself. And then look at all of these different results, the parcellation for one of them. Let's say this is the LEAP one, and this is the result for the HCP one. And then we want to look, for example, in this region, how similar are those between each other. So what we do is we compute, for example, the dice score, which is a measure of overlap, which means that if it's a perfect overlap, the value is 1. If they have no overlap at all, the value is 0. And then if we actually overlap them, we see that there are some voxels that are part of the one, but not part of the other. right? So we get a dice score that is not 0 because there's a lot of overlap, but it's also not 1 because there's some slight differences. And then the same thing we did comparing the actual volume of these regions of interest. For example, here we see this fairly thin region of interest, while we have a much broader mass here. So just counting the actual number, we see a difference in volume, and we can then compare to get like an estimate of percentage-wise how much larger or smaller is the actual region. And also because we want to get some idea about where the region is actually located in the brain, we compare these two. If we think about the center of gravity for this region, for example, we have a fairly big chunk here, fairly big chunk here. If we take the mean in terms of the center of gravity, we might end up somewhere here. If we take this for the other postulation, there's a big chunk here as well. So the center of gravity might actually be somewhere more up to the top left here, and then we can compute the actually Euclidean distance and voxel space between these 2 points. That gives us an idea of how actually the center of mass of these regions, how close to each other they actually are, whether or not they are distorted, as we've seen in the slides before, where we have these regions all of the sudden the subcortical area, so they are moved the structural connectome as well, and we computed network metrics of them. On one side, just these global average node strength, or the average connectivity each region has of the whole connectome, and also the clustering coefficient, giving us some idea of how well connected some local hubs are in the network. It was a basic graph theory metrics. And then we have this for each region individually, where we have the node strength again, how strong is this region connected to all other regions, and also how central is this region in terms of information processing. So if basically 2 regions talk to each other, do they have to go through this 3rd region or not, and how many times these shortest paths actually go through a specific region, to give some information about how central this is in their overall communication. And then we compare the differences here. And if we look at the global differences, we see not a lot of difference between the LEAP in yellow and the HCP processing results. So how similar they are to the ground truth. The dice scores, global dice scores, so if we take the full brain and compare those are fairly similar. Jacquard score, this similar overlap-based matrix, very highly correlated with dice. Volume differences, we find some differences, but not significant. The same goes for clustering and centrality of the nodes of the full brain. We see differences, but those are not significant. Only for the actual node strength, we see that the difference between ground truth and processing result is significantly smaller for the LEAP-based processing as compared to the HCP-based processing, where we see a lot of large differences in node strength between ground truth and the HCP result. This becomes a lot clearer if we actually look at the local differences. So when we see here, for example, if we take the dice score for each region of interest, again, on the x-axis, we have these rows of interest. We see that the dice score is on average always higher across all subjects for a specific region for LEAP, again, in yellow, as opposed to HCP in purple. And not only this, and not only is the line always higher, but also the variance is significantly smaller here as these error bars. So we have less variance in terms of the differences between ground truth and LEAP processing results, as opposed to the HCP, which kind of makes sense if we think about where these variance comes from. And this comes in particular from, for example, the lesion size. We can think of having very small subcortical lesions, not having a lot of impact on the processing, even if we don't mitigate for this lesion. But then again, if we think of these large lesions, for example, that we've seen before, we can think of very strong differences if we do not account for the lesion. So we have a lot of variance depending on what subject we're actually processing for the HCP data, and not so much for the LEAP, because we're actually mitigating the impact of those lesions. We see the same results for the distance from the center of gravity. So The distances are significantly larger if we do not account for the lesion. So if we just use the standard HCP processing as opposed to LEAP, and again, the variance also clearly larger. For distance, the same goes for the volume difference. So the counts of the actual region voxels, we see significantly increased for HCP as opposed to LEAP that is significantly closer to the ground truth. And also on the network metric level, we see the same thing. So the difference in node strength for each of those regions is significantly larger for HCP as opposed to LEAP, and the same for the node centrality as well. So if we want to look at this, what does this mean on the individual subject? Maybe grasp this a little bit better. It's like if we think about the subject again, where we imputed this lesion right here, and then if we look at the local distribution for each of those regions of the dice score, in this case, white means a dice score of 1, so there's no difference, and dark purple means a very low dice score, which means a very low agreement, which means strong differences. So that means that around this lesion, we see that the dice scores are actually very low, meaning there's a lot of difference between the ground truth and the HCP, as opposed to LEAP, where this is not as dominant. And the same goes for distance, for the center of gravity, as well as the volume difference. We see differences as well in LEAP, and these are some of these limitations that are still present in our mitigation measures, but they are more equally distributed and do not pertain so strongly to the actual impact of the lesion, as we've seen for those HCP processing results where we do not mitigate the actual lesion measures. And now that we have this validated and we have the processing pipeline done, to get back to actually TVB, what kind of research does this new processing pipeline facilitate? And this is, for example, one project that we're doing right now is we're looking into how can we better integrate lesion information into our structural connectome, into our tractography. So what we've seen before, what we implemented in our pipeline, and what has been the standard in the literature so far is we integrated the lesion as pathological tissue, and we said all of the tracks are basically like CSF, we ignored them. What we also did is we did not introduce this as pathological tissue and try to then look at the actual tracks that we end up with at the lesion. And what we see is that even though we do not specifically integrate lesion information in this tractography, we see that some of the topology of the lesion is well captured inside of the tracks that we capture. So we see this basically core of the lesion right here, still being reproduced. So we still have the spot where we, in fact, do not have tracks because of the damage the lesion caused at the core, and we still track some of the surrounding tracks. Even though they are technically part of the lesion, we still want to be able to capture them to then really look into what are the differences and how can we better relate those to, for example, a stroke biomarker. And this is informed by a nice study by some colleagues, Lippe in 2020. What they did is they looked at the actual lesion. We see this here down here is the T1 image, and we see the lesion. It's actually a MS lesion. Tuberous sclerosis is not a stroke lesion, but I think the principle applies still. And we see that these dark intensity values here inside of the white matter represent the lesion. So our lesion mask would include all of these dark voxels here. What this means is if we zoom in and then we look at the data, they look at the actual distribution that we mentioned before, like how probably are tracks to go along one direction in these voxels. We see that especially here at the outline of the lesions, at these slightly darker intensity values that are still part of our lesion mask, if we would draw it based on the T1 image, are actually identical to neighboring voxels. But we do still include them as pathological tissue and say all of this here is not, we don't want to track those. But we've also known, and we've seen this before, that the temporal dynamic of the lesion itself will most likely end up, at least in the stroke case, with a lesion that is still present here. As you've seen, these chronicle cores, they still pertain longitudinally, but all of those voxel will be back to their normal intensity values, so will be considered a healthy brain again. And this kind of supports this hypothesis if we actually look at the distribution, the probability distributions of those fiber orientations, they are still very much in line with neighboring voxels. So there can a case can be made to actually include these voxels in our tractography, because those, while there is captured in a T1 image, it doesn't necessarily translate to no tracks being captured here based on the fusion-weighted imaging. So there's a lot of ongoing research to try to get really at the bottom of how those recover and what does this recovery actually mean. And how we wanted to investigate this is we basically used TVB, and we thought about how do we embed this. So first of all, we had these two embedding measures or approaches doing tractography. So we said we have a lesion-based pathological tissue in anatomical tractography, and we don't. So we have two ways there of already creating structural connectomes. And then we looked at how do we then update the connectivity matrix that we load into TVB that we use as our the base for our digital twin. How can we update those? And we found three ways of saying, okay, first of all, we use the established approach, which has been done. There's already a lot of great stroke-specific brain network modeling literature out there, Falconel, Corbetta, etc. And the general approach there is still to say the region that is actually affected by the stroke is the weights are just set to 0. So we do not have the connection to the surrounding nodes anymore to kind of capture these abnormal activity patterns using brain network modeling. So that is our first approach. Our second approach was we actually went with the raw data that we get from the tractography, from our structured connectome, because we've already seen that it captures some of the topology of the lesion. So maybe there's also some information that we can get by not altering this, the raw structured connectivity matrix. And then the last approach was where we said, okay, we take the values that is given in our structured connectivity matrix and we update those with the information that we have about the lesion load. So where we say, if the lesion load is 100%, we weight the connectivity matrix 1 minus the lesion load. So then it will be 0 because the full region of interest is encapsulated by the actual lesion. So it's very likely that it is at the core of the lesion and will actually not recover over time. And then the more towards the outskirt of the lesion mass we go, the lower the lesion load will be. So it will just be a weighting of the actual connectivity value that we find. For example, if it's 50% covered, we will take the value and multiply it with 0.05. You get some additional embedding of lesion information into our TVB model. So what we end up with is basically 6 different structure connectivity matrices that is the base for our TVB model. And that we can then use to run our simulations. And if we look at them closely, we can see some differences. For example, here in the subcortical areas, we see that there's, if we set them to 0, all of those would be empty if they're touched by the lesion mass. So it's a very strong assumption. Here, we still see these regions present, but they're actually reduced because, for example, this region was 50% encapsulated by the lesion mass. Right? So we see these different embedding methods if we take a close look to the SC, but on a first look, because it is the same patient, that they look fairly similar. And what we did then is we ran a Stefanewski 3D model to create our simulations, and then we took a look at some interim grid parameter search to get the optimal parameter combination. So we see here is the correlation with our empirical data for a range of coupling parameters. This is the global coupling parameter and the conduction velocity speed that we varied here in our hyperparameter search of our TVB simulations. And just as a proof of concept, basically, is that we can see that even these slight differences in our TVB model and how we embed the actual lesion information significantly drives what parameter combination we get in TVB as these optimal combinations. So we see, if we think about it, these slight differences can come from small processing decisions down the line, and they will just be carried on across to our downstream analysis using TVB, where we have now different actual parameter combinations. So if we want then to use these parameters, these biophysical parameters, to actually infer something about the model, about the disease, we need to pay attention whether or not those have been driven by the processing and by the lesion embedding, or whether or not they have been driven by the actual disease, as opposed to the methodological bias that we introduce. just as an example for the type of research that if we pay close attention to the actual processing of our data, we can basically facilitate this full new field of research in TVB to better get to a point where we can get accurate brain network modeling to maybe use it to create new biomarkers for potential disease recovery or severity. And this is the talk part of today. And as I mentioned, I'm going to have a short sort of hands-on session. I put the material for this in the collab. I think you've seen this before, right? It was last week with Dennis in week 7. So I put a little bit of code and some data here. See the data as these three nifty images, and the code, which basically contains the Docker file and the registration experiment that we want to run. And I'm just going to go through this now. It's not really that we can do this at the same time because we need an actual container for this to be run. I hope this is readable. I zoomed in enough. So basically, if you take a first look here, this is our Docker file. As I mentioned, there's kind of these recipes to create the containers. It was initially built by the FSL development team, and we're using FSL to use the so-called FLIRT command, which is the linear rigid transformation registration algorithm in FSL, and also some utilities to get some infos about some of our data. And we just set up the container like this. It's a fairly simple approach. You can, if you want to use it, you can either download Docker and try to build it yourself. There are different ways of doing this. For example, you can build this container. This is all part of the registration experiment code that I put online as well. As you've seen before, the standard approach to actually building a container is just Docker build. You provide a tag that we want to use. In this case, I call it, it's part of my demos repository, and then FSL, and then we provide the actual Docker file that we use. Or if you don't want to build it yourself, you can, for example, pull it from my Docker Hub account if you have Docker already installed. And then there are 2 ways of running this. You can either start an interactive shell, which is what we're going to do now because we want to look at the actual steps, or you can directly run it by providing the code as well, the script that you want to run inside of the container. If you want to learn more about containers, I would suggest play around with this a little bit, download the code from the eBrains Colab and play around with it a little bit. And what we want to do now is basically want to take a little bit of a closer look at the most basic processing step, which was registration. To this end, we have this very nice brain image here that I shared with you on the Colab, and just because I always want to point out GDPR compliance, this is, and it's not anonymous data, right? Because it's brain data, it can never be anonymous. But this is actually my brain, so please don't share it with everybody. But feel free to play around with it a little bit on your PC. And we want to use this brain data and then use an atlas that we have to get my brain regions of interest. Actually, make this a bit larger so you can see better. And what we want to use is we want to use the HCP MMP1 atlas, so the multimodal parcellation based on Glasser. And if we see this right here, we have the image already, HCP MMP1 in MNI 152, so basically in template space. And what this means is if I actually overlay this data, we see it does not fit my brain. And this is exactly what I mentioned before during the registration discussion, is that those live in totally different spaces. The atlas itself is in MNI space based on 152 healthy controls, averaged into the same space. And this is my individual space, right? So they are not mine. So the goal now is to get them to overlap so that we can use this atlas for my brain, which is not how this type of analysis is done anymore these days, but it's basically this classic introductory approach to trying to get some information based on regions of interest into a subject space. And the way we do this is first we start our container here. I already have it. And now we can see it actually changed from my desktop PC. We are now as a root user inside of the container, and we can run all the functionality that we have available. And because I also provided this -v again, so the volume that I'm mounting, which is exactly where I was, my working directory, and the data folder, which is exactly this processing and then the data folder. So if I now go into cd-data and let's say, what do we have? We see exactly these three images. So what we want to do is we first define some variables. So input is obviously our input data. Our reference will be an actual template of the MNI space. This is where we want to reference it to. And our atlas is the atlas that we've just seen. If we actually look at this data again, if we now look at the MNI space, because we want to register our atlas to my personal brain or my personal brain to the atlas, if I try to register those, we will have the same problem that we had during when we discussed cost function masking. The sheer sizes are so different that the optimization problem will blow up the parcellation to try to match it and minimize them. But what we can do is we also have an actual brain in MNI space that we see here. So the idea now is this we can register to my brain because it even has skull, it has like a full brain available as opposed to only the atlas. And we can see if we here in the MNI template, we overlay the atlas, we see a perfect alignment. And this is basically what we want to have for my individual brain and not for the template brain anymore. So the idea is that we register this image to my brain and then apply it to the atlas. So to do this, we define these input variables, we define our output file names, and we can also start with getting just some information about our inputs themselves. So FSL info gives you some good idea, for example, the dimensionality. So this is the resolution of our image, of my input image. It's 192 by 256, 256. And we compare this to our MNI space. We see it's actually significantly smaller. So we already see a large difference in resolution of the images. Ideally, this will be overcome by our registration, the way we use it with the default values. If you want to learn a little bit about Flirt itself, you can already just call the help function, and we see a whole range of available options. Right? We have this basic structure of just providing an input, a reference, and then how it is saved. But we already see a lot of these variables that define specific aspects of this optimization problem. For example, here, cost is exactly the cost function masking that I mentioned before, where we have a whole range of them available, the default being a core ratio, but also mutual information, least square, so whatever function we want to use to compute those differences between the images that we want to warp towards each other. So we see there's already a lot of information that can be used to change slightly the actual registration program. And to first do this, we just run this command. I'm going to start it already because it will take like 20 seconds or so. And what we provide is we provide our input image, we provide our reference image that we want to. So my brain that is mapped into the reference space into the MNI. And then we also save the transformation matrix. So the information that needs to be applied to each voxel to go from my input space to the reference space and the actual output image to see whether or not the registration worked. And we're now actually aligned with the template space as we wanted it to. And hopefully this will be done in a few seconds. So we just spent some awkward silence together once this is done. I think it took like 15 seconds or so when I did it previously. There we go. And now we see a new image here that we called input to ref. So if this is basically my image now mapped into reference space. And if we look at it right here, we already see a difference in terms of the smoothness of the image because it got registered to the template space, which was like this really, if we look at it again, abnormally smooth. This is not a real brain, right? This is this average that we can see. So this was mapped into the average template space. And if we now overlap the atlas to it, we can see it's actually aligning with our brain as opposed to before. We see here, for example, these areas where there's not a lot of good alignment, or we have these actual holes in the parcellation, etc. This is why I mentioned that these days, this is not normally the normal approach anymore, but it was classically used to just get some idea about these regions in my individual brain. to make things a little bit clearer in terms of what actually these processing steps, now these variables, how they can influence the registration. We now going to use a different way. We're going to do the same thing again, but we now going to use a different interpolation method. We've just seen the one image that all of a sudden got really smooth, because now this change in resolution basically resulted in, for example, 4 voxels now being all mapped into a single voxel because of the lower resolution that we have. And the question then comes into what intensity value does this voxel get? And the default interpolation method that is used here, we used before in Floyd, was a trilinear interpolation. So it kind of creates a new intensity value based on the interpolation, on the trilinear relationship between the actual voxel that make up this new voxel. And now we're going to use a different one, and we're going to say, actually, the interpolation method that we want to use now is nearest neighbor. So it's not going to be, so we're just going to run it again, because it takes some seconds. So the interpolation now is not going to create new intensity values anymore to make it fit, but rather it's going to use intensity values that are already present in the image. So the nearest neighboring voxel will then be used to make up the new voxel, and that is closest to it, right? So it's a little bit more of a clustering of already existing intensity values, as opposed to creating these new intensity values based on the trilinear interpolation that was the default that we used for Flirt. And I can already prepare the comparison. So this was the image that was originally created, and now we wait a couple of seconds until the new one is created, which will then be called interref_NN. There we go. And if we now look at the comparison between those two, we already see that this is significantly less smooth on the right compared to the left, right? Especially if we actually remove smooth as for the visualization, we see that the actual intensity values that we see here are not as smooth as the values that we find here. So we already see here this one simple parameter in one registration call already creates this input, this difference in registration results. And we've noticed before, right, if we talked about surface extraction, segmentation, how these single voxel values, the actual values of those, matter to define where borders are, where surfaces, etc. So we can think of these small steps actually being carried on again across the whole processing pipeline and up to the analysis, the downstream analysis at the end, to really have an impact and to really be something that we need to be aware in terms of where group differences, for example, might come from. And as a last step, we can actually also just compute the difference here. I forgot about this. That creates the difference image between the two registered images that we created, between in2ref and in2ref with the nearest neighbor. And if we look at this, we already see that there is a huge difference. It's basically recreating the whole skull, and also we can see foldings, the actual cortex here being reproduced, which means that this is where we find already differences just by this single parameter to the registration call that we're doing. Just to really kind of highlight the importance of paying attention to these if we want to create our own digital twins. And what we want to do now is something that we also did and that we discussed quickly when we talked about the visual brain transplant, where we had mirror images and we moved data back and forth to left tilted, right tilted, and then the mirror, and then just taking half of this transformation matrix. So just to get some idea what we can do with this, we're now actually going to invert the registration. So we had this from our input space to the reference space, right? We put my brain into the M&I template using the nearest neighbor method here, and then we can convert it by just saying we convert this input matrix that we saved, and we now turn it into reference towards input as opposed to input towards reference. And then we can apply this here at the end to the actual atlas, which means that now we're, instead of making my brain fit the M&I template, we're going to take the M&I based atlas and make it fit my brain. This is fairly quick. And now if we actually do this again, look at the original input image here that we see, and we overlay this again with the with the new atlas that we find. Where is that atlas? Should be HCP subject. Was it not created? Here, let me check if there's something I did. Oh yeah, I did something wrong. I did not define the variable to save it. There we go. So now if we take this, the original input image, and we overlay the HCP subject, we see that now it is in my space, as opposed to before where we had it being in a totally different space, someplace totally else. Again, of course, it's not a perfect match, but it's a close enough match given for how simple a registration step we did here. Just play around. So I would suggest, if you're interested in this, and if you want to really pay attention to your digital twins, especially we talk a lot about getting TVB into the clinical practice, right? Getting into these clinical use cases, we want to make really sure that the data that we have is accurately representing our disease, our subject, etc., so that our TVB models, our digital twins, are accurate representation of the empirical data, and are not biased by the actual processing that we're doing along the way. And I think with this, I'm basically done. So I don't know if there were any questions in between. I just see there's a chat, but I don't know if this is an actual question. Nope, that's the link. Thanks, Judy. So I guess I just stop sharing, and if there are questions, happy to answer them. Otherwise, I guess it's a Tuesday evening for everybody.